So hello, this is Violeta Munoz and Anna Farian from Safoso, and we are excited today to hear from Steven Sarrazin and Philip Joosten from University of Ghent, what they've been working on in the last couple of years and months in the frame of the work package 5 of the AMR project. So Philip Joosten is a PhD student and Steven Sarrazin is a postdoc at the Veterinary Epidemiology unit at the Faculty of Veterinary Medicine at the University of Ghent. Hi, both of you.
Hi, hello.
Hi, thank you for an introduction.
At the Veterinary Epidemiology unit of Ghent University, each of us is working on the triangle between antimicrobial resistance, antimicrobial usage, and biosecurity as a part of disease prevention. And this area of expertise fits very well within the EFFORT project and more specifically Work Package 5, where we are Work Package 2. Because within this work package, in the first part, we described antimicrobial usage and biosecurity at the herd level for several animal species, for instance, broilers and pigs. And in the second part, we want to link it to antimicrobial resistance and also try to identify which are drivers for antimicrobial usage. And now, Philippe will present some of the most interesting results we have so far, and basically these are on antimicrobial usage.
So when we did the analysis for the antimicrobial usage data, we plotted the usage over one production round, as you can see here in this figure for the broilers. And that's on the x-axis, and on the y-axis you see the percentage of farms that is using antimicrobials. So we saw that in all species that there is a higher usage in the beginning of production, and here this is shown by the box in the first week of production, and this box represents 47% of the total usage in broilers during the production rounds we sampled within EFFORT. And this pattern of high usage in the beginning of production is something that we see over all species. So for veal calves, for turkeys, but also for the pig industry. And then, based on our quantification, we can express usage in over time of one animal. So let's say a broiler is reared for 42 days, and our quantification ends up with a number of 10%, and that broiler is being treated 10% of that of its life expectancy. So when we compare this for all the species that we sampled within EFFORT, we see that on average these animals are being treated with antimicrobials during 10% of their life, with small variations between species, so ranging from 9 for the pig industry and for the broilers towards 13% within the veal calf industry. This might not seem that much, but if we compare this, if we would go to human level, this would mean that we as humans would be taking antimicrobials during 1 month each year of our lives. So that's quite a substantial amount of antimicrobials if you compare it like this. And then finally, most important for the One Health perspective is the usage of the critically important antimicrobials, and there we see that these are still frequently used within these different types of animal production systems. So for each species we have the 3 biggest antimicrobial classes listed within EFFORT, and the blue ones are the ones that are on the critically important antimicrobial list of the WHO. And then for each species, these are 2 classes each time that are on the list and represent, let's say, on average 30 to 40% of the total usage registered within EFFORT for that species.
Now that we have the data on antimicrobial usage and biosecurity, It is interesting to try to link those results to antimicrobial resistance. Within the effort project, antimicrobial resistance has been determined in several ways. Some of us have been using metagenomics, but antimicrobial resistance has also been determined by using minimum inhibitory concentrations for E. coli isolates. And some results on broilers have been shown that there is a positive association between the usage of aminopenicillins, such as ampicillin, and the likelihood to be resistant for ampicillin. So the higher the usage of aminopenicillins, the more likely an E. coli isolate was resistant to ampicillin. Also, we found that a higher biosecurity for the introduction of day-old chicks and animals leaving the farm was negatively associated with strains being resistant to ampicillin. So the higher the biosecurity, the less likely strains were resistant to ampicillin. More results on the link between usage, biosecurity, and AMR will follow.
So based on these studies and other results of the effort project, we hope to translate these findings into an advice on herd level regarding antimicrobial usage and biosecurity. A good example of how we could do this is the biocheck that has been developed by our veterinary epidemiology unit at the University of Ghent. That is a free online tool for farmers where they can fill in a questionnaire that scores their biosecurity on herd level. And this gives them a feedback where they get an advice to how they can improve their biosecurity on farm level. And it's tools like this that can offer farmers a way to improve their biosecurity and therefore also maybe reduce their antimicrobial usage.
Some of the results have already been disseminated. In the end of February, the first results on antimicrobial usage in pigs and broilers have been presented during the Acting Conference in Ghent. The end of March, Philip and I went to Estonia to present data on usage for broilers and pigs during the FP7 conference. And Philip submitted several abstracts for the ISFI conference at the end of this year. Furthermore, we have the first papers in preparation to be submitted quite soon on usage in broilers and pigs. But as said before, many more results will follow on the link between usage and resistance, biosecurity and resistance, and also the use in other species.